Today, we welcome our listeners to become genome explorers. We're going to make your DNA dance with excitement and your genes jitter with anticipation. We are diving headfirst into the world of advanced therapies.
Picture this, a world where your genetic code isn't just a blueprint, but it's a treasure map that leads to treatment tailored to fit you and your disease perfectly. We're not talking about the future, folks. We are talking about right now.
So we will cover current progress in advanced therapies. We're going to talk about the challenges, the opportunities in seamlessly integrating genomic medicine into everyday healthcare. Finally, we're going to look ahead and see what's next for this exciting field.
Advanced therapies are what matter today on Discovery Matters.
When we refer to advanced therapies, we mean medicines that are based on genes or tissues or cells, and they are different from the antibody or protein-based biologics we know well. For context, there are around 1,100 active clinical trials of advanced therapies, but only around 20 advanced therapy products have been approved and are commercially available so far. That is a statistic from early in the year 2024, according to Nature Review. Check the show notes if you want to see that full report.
To unravel what actually needs to be done to get more of these advanced therapies or genomic medicines approved, let's get some grounding in the progress that's been made so far.
Absolutely. And our podcast longtime listeners will recognize Daria Donati. If you're new to the podcast, then Daria is our very own Chief Scientific Officer of Genomic Medicine here at Cytiva. And she was talking about 2023 as being a critical year for this field.
In the last 5 years, between 2017 and 2022, 5 different gene therapies have been approved. Only in 2023, 5 other therapies in the genomic medicine space have been approved. And that's pretty amazing. It gives really the feeling of the moment we are living and the speed in which genomic medicine is being recognized globally by the regulatory authorities. That has been followed now in January by the first approval of one-time treatment for the first solid tumor, that is melanoma with infiltrated lymphocytes that are TILs. And not only that, by a statement from Peter Marks, one of the FDA top officials overseeing the cell and gene therapy space, saying that accelerated approval will become the norm in the gene therapy space, especially for rare diseases, for these people that have one shot in life to get a treatment for a disease that otherwise will terminate their life.
Now, that's rapid progress, and it's such an exciting time for the industry. But are these therapies actually getting into everyday use where they benefit patients?
Simple answer, yes. Currently, we have yet to seamlessly blend genomic medicine into our healthcare system. So to do so, we actually need to unleash diverse technologies.
We are witnessing a breakthrough scientific moment where a lot of discoveries are brought to the application in the clinic. Millions and millions of people have been treated with the first RNA therapy. The vaccine was for the first time delivered by a lipid nanoparticle that in this case deliver RNA, but it can be used to deliver engineering tools like the CRISPR-Cas complex or DNAs. So there are a lot of different technologies that are coming to life, however they live in pockets. So they are restricted to specific areas. So what do we really need to help the genomic medicine to become part of the everyday treatment and life, the way we make these therapies, the way we produce cell therapies? And what would that enable? First, we'll enable the reduction of the cost. With the reduction of the cost and the simplification of how you bring them to life and to the patient, suddenly you increase accessibility. So when I think really about the challenges we have in a pocket of technologies that are still living separate life that need to be gathered, and then enabling this change in real life, we have the unique opportunity to bring together and generate synergies between technologies or application. This is really the unique opportunity to enable a leapfrog and acceleration of innovation in the genomic medicine space.
To break these pockets and enable technologies is a strong vision. We can all get behind it. It's pretty non-controversial, but it needs motivation and dedication.
Sometimes working on developing an hardware that enables a workflow makes you feel so far away from the patients. But with the time, we are getting closer and closer to the patient. We really see how a new formulation in a delivery system, as much as crashing a workflow, reducing and simplifying, has really an impact on the life of a patient or 10 patients or 15 patients, actually, even more. The other exciting part of all this story is that we are increasingly not only looking inside, but looking outside to bring expertise. We are working with the best scientists in the world to further develop our own technology, marrying the best of the external technologies with all the industrial knowledge that we have.
To further glean what is happening in genomic medicine, let's focus on the foundation of all genomic medicines, cells. As the fundamental units of life and the carriers of our DNA, they're the foundation to understand genetic information, to regulate gene expression, to study how disease works, develop new therapies, and to put personalized treatment to work for patients.
This has been a transition to using cells as medicine. We've been doing that for a long time with bone marrow transplantation. We've been using cells as medicine and just transplanting just normal cells, unmanipulated. CAR-T was how can we make those cells do something that they normally wouldn't do? You're putting essentially what's an antibody, which is secreted by B cells, on the surface of a T cell so that it can then go recognize a tumor. T cells weren't designed to do that. The original ways to do this have been through using viruses, using lentiviruses or retroviruses to actually insert that capability into the gene. And they go in rather randomly. Viruses go in, you get integration, and then it expresses the CAR. And so now the advancement with CRISPR is you're using these molecular scissors that you're directing exactly where it is you want to snip. It's not dependent on a viral integration event to do that. People still use viruses. In some cases have had to make gene editing work, but it's an evolution towards precision of our ability to manipulate the genome in ways that are beneficial.
That's Jen Moody, the Vice President of External Innovation of Genomic Medicines for Danaher, our parent company. Jen has seen the transition from the use of viruses and CAR-T advance like precision engineering.
She did her PhD on hematopoietic stem cells, B cells, and T cells and mouse models.
After my postdoc, very conscious decision on my part, I joined a company called Stem Cell Technologies. The timing on that was really interesting because I was always really interested in pluripotent stem cells. And what I didn't know is that in the months where I was interviewing, they were just closing a deal where they were signing a license agreement for the first feeder-free pluripotent stem cell media. Because I had said that I wanted to do that, that ended up being my first product. That product became a juggernaut. We put it out to the world, and it changed the way the world grew those cells. And it enabled the whole field. And the amount of pluripotent stem cell research and how that's advanced in the time since we launched that media, I feel like I had a really tangible part in that.
Let's get grounded a second. I mean, cell therapy, gene therapy, genomic medicine, how are all these terms linked?
Genomic medicine, it encompasses cell therapy, gene therapy, and gene editing, nucleic acid therapy. What all three of those things have in common are cells. You're manipulating cells. You also manipulate stem cells with and use them for cell therapy for gene therapy and for gene editing. Stem cells can have an additional layer of what is generally referred to as regenerative medicine. So it is replacing gene function without necessarily having to manipulate them, using the stem cells to create cells to replace gene function, as opposed to CAR T, where you take the cells out, manipulate them, put them back in, or even in vivo CAR T or in vivo gene editing, where you're targeting cells in the body to do it. The stem cell piece is you're harnessing these pluripotent stem cells. You're harvesting them to create the cell types in vitro that you can then put in. And then you use those for replacing cells that have lost their function.
Hey, Dodie, before we move on to the next section, shall we just interrupt the podcast for a second and do a couple of adverts? You know how podcasts are sometimes funded by adverts?
Then let's be clear, these are not organizations that have given us money. These are organizations that we work with and we just want to talk about because we think they're wonderful.
Exactly. They're lovely people, and we thought we might use our reach to help get their stories to you.
So here's one. I'm going to tell you about the N. Lorem Foundation. The N. Lorem Foundation pioneers treatment for nano-rare patients using cutting-edge ASO technology, providing lifelong care free of charge. Dive into their Patient Empowerment Program podcast for insightful interviews and lessons spanning basic science to drug development, aimed at fostering a supportive community for these patients.
Thank you, Dodie. Let's go back to the podcast.
Stem cells' regenerative power combines with genomics genetic insights. Stem cells are nature's building blocks. Genomics reveals the genetic blueprints, like that treasure map that you talked about, Connor. Understanding genes lets us program stem cells as tailored treatments, repairing damage personalized to each patient. Genomic medicine harnesses stem cells to tailor therapies for each patient.
So we have the building blocks, we have the instruction manual. All we need now is the effective design.
But first we need to understand the risks more fully. Jen is speaking about cars here, but the reality is that all these therapies are on the frontier and we're encountering hurdles as we go.
Sometimes you have this risk of secondary cancers. We don't know whether that's related to the car necessarily all the time, or there's lots of different reasons why that might happen. And so they put this black box warning. It tempers things a little bit. Because at the end of the day, this is biology. And as much as you think you can control all of biology, and biology will teach you over time that you don't know everything that you think you know. Obviously, the benefit outweighs the risks here, and the FDA has been very clear about that. There's lots of room for improvements when it comes to safety. People are tinkering with different ways of delivering. Is it the viruses that are used? Should you be using non-viral method to deliver and make your cars? There's talk about stained expression of a CAR like you would get from a virus. So only expressing it via mRNA so that it's only transient, and exploring whether that's better and for what indications that might be better. There's talk about traditionally all of these have been done ex vivo, these autologous therapies, ex vivo, where you take the cells out, you manipulate them, you put them back in. Can we be more specific? Can you do it in vivo? But then there's also these manufacturing considerations that you think about, not just from how we manufacture the product in terms of efficiency. They're more armored, borrowing from cancer cells to put these mutations in that don't make a T-cell tumor, but it makes it so it doesn't get exhausted.
Despite this, what role will genomic medicine assume in the near term, say within the next 5 to 10 years? And how might the role evolve even further into the future?
We're changing the practice of medicine in the 5 to 10 year frame. You're going to see traction on things like solid tumors. You're going to see traction on things like addressing autoimmunity, not just cancer, with CARs. It's going to start to broaden. There's going to be approved therapies for things outside of oncology, even for gene editing, for things outside of these diseases that we're tackling. I think we're going to look back, and the things that we're solving today that we think are so miraculous, we're going to look back and see that, oh, that was low-hanging fruit. And one thing that I'm really curious about, how AI is going to thread itself through this, from protein engineering to people talking about AI eliminating placebo arms by modeling. And it's spanning everything, manufacturability, just everything is being touched by AI. But suffice to say, the practice of medicine will be fundamentally changed.
Before we get too ahead of ourselves, to see this vision become a reality, we need to reduce manufacturing hurdles to get these therapies to patients faster. So here's Demaris Mills, Group Executive of Genomic Medicines at IDT, another one of our sister Danaher companies. And Connor, I believe you know her.
I do. We've met.
We see the promise of genomic medicine. We see that we are now coming to market with cures for the previously incurable. But when I look at what does it mean to actually operationalize that, to provide these treatments to patients and actually get that patient access, we have a very long way to go. There are hurdles at every step of the manufacturing process of these genomic medicines. And then there are regulatory hurdles. For example, in the case of gene-edited cures, we might identify a solution, but because of the time that it takes to move through the traditional regulatory process does not enable that cure to get to that patient. It points out that from scientific to manufacturing to regulatory processes, those processes are designed around very different set of treatments. And we need to really rethink the way that we operationalize and make these treatments accessible.
To get to the stage of integrating genomic medicine into everyday healthcare, we need to rethink so much of everything, from the manufacturing to the regulatory processes.
How do we get ahead of that? How do we accelerate things? What can we invest in, build, or do now that could really accelerate the process of bringing these therapies to market quickly?
I actually think that to address the biggest pain points or the biggest barriers that are preventing us from getting these treatments to markets and to patients, that is because of the sheer breadth of technologies that we have. If you were to lay across the manufacturing workflow, the different capabilities actually span that workflow quite nicely. And with not just with mediocre capability, with truly differentiated technologies, in many cases gold standard or best of breed technologies to be able to solve for these problems, it's really incumbent upon us to bring those pieces together in a way that solves for the acute problems that our customers face.
Remember what Daria said at the top of the episode. We need to simplify workflows and bring technologies together rather than have them situated in pockets. To take advantage of these opportunities and address the challenges that Demaris was talking about, what do you suppose is critical?
We say it so often on this podcast that collaboration is important, but here what actually moves the needle is collaborating with intent.
So I think you're going to be delighted to meet our next guest, Fyodor Urnov. He is a world-famous biomedical researcher. He has played such a leading role in the field of genome editing. Fyodor is a professor of molecular and cell biology at the University of California, Berkeley, and the Director of Technology and Translation at the Innovative Genomics Institute, the IGI.
I'm going to temper my fanboy tendencies here with just a public service announcement on full disclosure. So Cytiva and its sister companies in Danaher are collaborating with the IGI to clear the obstacles that stand between rare disease patients and these very exciting new, safe, effective therapies.
Connor, if you don't mind, I actually heard Fyodor describe the IGI in such a great way, so let's turn it over to Fyodor to tell us more.
The dream is to make sure that CRISPR, metaphorically speaking, spreads its wings in the maximum possible beneficial way to impact all aspects of society, building genetic engineering tools to build better medicines. We are logistically not just a lovely building on the UC Berkeley campus. We are a joint effort with the University of California, San Francisco, which is across the bay from us. They have not just a medical school, but one of the world's greatest teaching hospitals and a large cohort of physicians and physician scientists who specialize in taking on really challenging diseases, including the ones that we work on with them. It's also a research operation that brings together faculty from multiple different departments here at UC Berkeley with the overall principle that we form non-overlapping facets of the same solution. We have bioengineers building delivery tools, and we have biochemists like Jennifer Doudna figuring out how Cas9 works at an atomic level. And then we have translational people like myself who know how to take a CRISPR and a delivery tool and then put it together and make it into something that could be used as a therapy.
Talk about your scientific A-teams, am I right? There's a major need for this delivery tool. So, for example, there's a CRISPR cure approved for sickle cell disease. That is a heroic achievement. There are 120,000 people in the United States at last count with that disease, millions worldwide, 300 million people with genetic diseases that in principle could be treatable via CRISPR.
Let's go back to the building blocks analogy, and IGI is pulling in all sorts of building blocks into bringing these therapies to these patients who really need them, right?
If I closed my eyes and started weaving a quilt.
There are a lot of steps involved which might partially explain why the first CRISPR therapy was approved just last year. Casgevy is designed to treat two debilitating blood disorders, sickle cell disease and transfusion-dependent beta thalassemia. And these lifelong genetic disorders are difficult to manage for patients. But it's not just about the number of steps involved, those building blocks. It's about converging techniques, experience, knowledge, CRISPR, nucleic acids in this instance, and it takes a breadth of knowledge to put them together in a safe, scalable way. So the goal is to combine all of this to deliver an outcome rather than create a specific type of therapy.
The patient is the real judge of whether what we've built is safe. Ultimately, we have a very specific goal. In 3 years, we would like to treat a child at UCSF with HLH. And just a few short months after, our goal is to treat a child with Artemis SCID at UCSF. And people will say, Fyodor, wait, you have to wait this long? Yes, because remember, we are currently functioning in a system, the framework of which was built back in 2010. We have a 15-year-old regulatory and manufacturing and de-risking environment. So our first foray into whether it's HLH or Artemis SCID will have to take not just this amount of time, but also because we're innovating. The key idea, though, is once we're done with that, then bringing in the next such condition, the next condition where the immune system is hyperactive, or the next severe combined immune deficiency will not take 4 years, but will take 6 months.
HLH is a rare disease that usually occurs in infants and young children and affects the immune system, causing abnormal blood cells to accumulate in the spleen and liver. It can be inherited or acquired, and it can be treated with drugs or stem cell transplant.
This is very optimistic, but if one isn't an optimist in our field, then one works on something else. We have to have confidence in our abilities, in the technologies, and frankly, in the fact that there is this thing called 1 + 1 = 7. There is non-additive synergy between strengths. And we are absolutely convinced that the teams at UCSF and UCLA and the IGI on the one hand, and the teams at the Danaher operating companies are precisely an example of this 1 + 1 = 7 non-additive synergy.
So we're talking about diseases that are triggered by genetic faults and genetic makeup of individuals. What about what Daria said earlier, that we have to make genomic medicine part of our everyday health and life? What about common illnesses like the cold?
Well, CRISPR is slowly but surely making forays into common disease.
Let's be clear. Genetic diseases are devastating and an enormous area of unmet need, but they're not the leading killers. Thinking about chronic obstructive pulmonary disease, or fibrosis, pulmonary fibrosis, in thinking about the lung. And people will say, but wait a minute, these are not genetic. They are. The genetics are complex, but that does not mean that you cannot chart a path where the right kind of CRISPR engineering could not give you a clinically beneficial outcome. And again, the example of cardiovascular disease, where several gene editing biotechs are going after one specific gene with the ultimate goal of building a therapeutic for cardiovascular disease more broadly, is a good one. So my dream beyond genetic disease is ultimately that, frankly, big pharma will learn from our clinical track record and from, frankly, the visibility of our effort that there is a there in this CRISPR thing. And CRISPR is remarkable, right? Think about biologics. Once upon a time, that wasn't even a class. And look at what's happening today. Think about CAR T. 30,000 patients have been treated with CAR T in 10 short years. It's kind of astonishing, right? My huge dream for our technology is that we will pave the way for CRISPR as a therapeutic for a larger segment of diseases. That, as I said, will require big pharma, which is exactly how it should be. And I'm hopeful that our effort will say to companies of a scale that can take on something like inflammatory bowel disease or COPD, the genetic evidence for rare disorders in this space is pretty compelling. Let's see what we can do in taking on the more common forms.
There is a long way to go, but this work is really very, very exciting.
Fyodor had this fantastic illustration of what his work is like, and he compared it to when Michelangelo saw the block of marble and he knew that the David was within that block of marble.
As we're building a streamlined path to CRISPR cures, we kind of see the shape of that thing inside the large unwieldy thing in front of us. So we basically take away the unnecessary to discover what's in there. But what does success look like? How do we define a win? And yes, of course, we want to build a platform to cure multiple people. At the Academia in Florence, you see David, which is this endlessly transcendent achievement of the human spirit. But there are also these unfinished slaves, where basically slabs of marble, where Michelangelo chiseled out the overall outline, maybe an arm or a head or a shoulder or a leg, and then his interest turned elsewhere. Ultimately, it's so inspirational to think about the fact that little bits and pieces of our innovation ultimately will be useful. So that the big quilt we're building to try to provide cures, ultimately little facets of that quilt could also be useful as they enable broader fields of genetic medicine.
Our journey into genomic medicine began with looking at what's happening now and how advanced therapies are shaping up. While there have been notable advancements, this episode also acknowledges the challenges in seamlessly integrating genomic medicine or advanced therapies into everyday healthcare. Our scientists, experts emphasize the need to overcome hurdles in technology integration, manufacturing processes, and regulatory frameworks to make these therapies more accessible and widespread.
So despite the challenges, we're optimistic about the future of these therapies. Just underscores how important collaboration and technological innovation and the understanding of biology and long-term strategic planning and thinking about the regulation is to driving the field forward. And addressing all of these barriers and using the technologies that we have and thinking about what we might have in the future, we can build a shared vision of realizing the value of this progress for human health and turning it into a fundamental part of how we treat healthcare in the future. Super, super exciting. And of course, it means that for everyone in this field, every day is a school day, Dodi. So what have you learned this week?
Well, it's early April when we're recording this. I had to rifle through a lot of April Fool's jokes on some of my favorite science websites like ScienceDaily.com and Science.org, and there were a few. But this article was published a few days before the 1st of April, so I'm pretty sure it's real. And it is about scientists who did some genetic analysis of 2 teeth that were found in a cave in Ireland. And you'll be delighted to hear about this because they could, with genetic analysis, they could analyze the impact of how our diets have changed. And it also revealed a lot about this particular man's microbiome.
Oh, wow. What, like in his teeth? Extraordinary.
In his teeth. Yes, exactly.
He obviously didn't brush.
He didn't. There weren't teeth whiteners back in the day.
Yeah, but they did just a stick, like a toothstick or something, right? That must mean something.
Yeah, some sort of grain or something. But Interestingly, the team of researchers found evidence to support this disappearing microbiome hypothesis. So you'll understand that, right? So we're talking about the loss of biodiversity. And so the Bronze Age teeth produced different strains of a bacteria that has been identified as one of the causes of gum disease. I'm not even going to, Tannerella forsythia.
Well, I think that sounds like you nailed it first time. You tried to pronounce that one.
I hope so. Mm-hmm. And the PhD candidate at Trinity, I'm going to say the student's surname is Jackson. I'm not even going to venture a guess about the first name. It looks like Isolt. But says that these strains from a single ancient mouth were more genetically different from one another than any pair of modern strains in the data set that they currently have, despite modern samples deriving from, listen to this range, Europe, Japan, and the USA. So they're finding that what a loss in biodiversity, and we need to understand that better. All of that by looking at the genes from 2 teeth from 4,000 years ago.
Teeth.
Awesome.
And that's extraordinary how actually looking at things from the past allows us insight into how the world has changed. And you know how we like serendipity? We have a bit of serendipity going on here, because what I learned today is also about looking at things from the past to understand what's going on today. So we're not delving into an Irish bog here. We are diving into the deep blue sea, and believe it or not, we're searching for decades-old cans of salmon. So it's not like a bizarre treasure hunt. It's like an intriguing piece of detective work where old tins of salmon have become an unexpected source of insight into the health of our oceans. And researchers...
Should I ask what those tins of salmon are doing in the ocean, or we're going to move right on past that?
I don't know where they got the old tins of salmon. They maybe got them off a ship or a boat, or I hope they didn't get them from anyone's cupboard. And what they've done is researchers at the University of Washington, led by a parasite ecologist, came...
At Seattle? At the UW?
UW, your alma mater.
Yes.
They came up with a novel way to kind of look at the historical shifts in marine parasite populations. They used salmon as an indicator species. Super clever, this. The salmon, specifically the pre-packaged salmon that come in cans, the ones swimming around in the Pacific Northwest in tin cans, they carry certain parasites that complete their life cycle in larger marine predators such as seals and killer whales. So the level of infection in salmon directly reflects the level of infection in their predators. So where do you find the old salmon? Well, you find them from an adventure that sounds like it's straight out of a movie. The Seafood Products Association was clearing out its basement full of long expired canned salmon, and they offered these cans to the researchers. Their hunch paid off, and dissecting the salmon, they found that many of the cans were just brimming with parasites that provided an unexpected historical record. And they discovered that over 42 years, infection rates had increased in 2 of the 4 salmon species that they were looking at, and they suspected that this might indicate a conservation success story. So it might seem unappealing, but an uptick in marine parasites could be a sign that marine mammal populations are bouncing back, and that echoes similar findings in other parts of the world. So
that's a hopeful note.
A hopeful note and a wonderful sort of coming together of all sorts of research areas.
But let's say this, don't try that at home.
Yeah, don't eat the salmon mousse.
Do not go digging in the back of your pantry.
Just be careful. Eat within the consume by date, folks. It's good advice. And if it goes to waste, maybe there's a researcher somewhere who wants to dig into it. So...
Our producer is Beth Armit Brewster, editing, mixing, supervision by Banda Productions, music from Epidemic Sound. Thank you for listening. My name is Dodie Axelson.
My name is Conor McKechnie. Make sure you rate us on Spotify or whichever platform you use. We'll see you when we come back with another episode of Discovery Matters.